Hi there, welcome to section 2 of the 2025 National 5 Biology paper. We're going to go through each of the questions in section 2 as well and just check the answers, but also explain where it's needed. So question 1A, students carried out an investigation to compare the ultrastructure of typical bacterial and fungal cells. Part 1, the table shows their results for the fungal cells. You have to complete the column for the bacterial cell by placing a tick in the appropriate boxes. So for the bacterial cell, we know that there are three cells that have got a cell wall: bacterial cells, fungal cells, and plant cells. So we can tick that one. We know that the bacterial cell is the only cell without a nucleus, so we're going to leave that blank. We know that bacterial cells don't have any membrane-bound organelles, i.e. any organelles that have a membrane, which a mitochondria does, so it does not have that. It has ribosomes because it does need to produce some proteins. It is the only cell which has a plasmid, and like all the other cells, it has a cell membrane. However, unlike the fungal and plant cells, it does not have a vacuole. Okay, so that's what we're looking for there. Those are fairly straightforward. They should be ones that you have memorized from your notes in terms of the structures of all the various cells and know which cells have which structures. Part 2, the structure of the cell wall in the fungal cell was found to be different to that of a plant cell. Name the structural carbohydrate that makes up a plant cell wall. We also learn this in the photosynthesis topic in 3.3, that the carbohydrate that's made for cell walls is cellulose. Okay, it also mentions it in topic 1.1, and it says simply that the bacterial and fungal cell are made of different materials, but the plant cell wall is made of cellulose. So that's another one where you needed to know that. That was a knowledge-based question there. 1B, on average, muscle cells contain 2,500 mitochondria, liver cells contain 2,000 mitochondria, and cheek cells can have 200. You have to calculate the simple whole number ratio of the number of mitochondria in these cells. Okay, so the first thing for this question that you should do is look at the way they want the answer displayed in terms of the ratio, and then write down your initial numbers in that order. Okay, so step 1, we'll get the numbers written down, the raw numbers that the question have given us. So that's 2,500, 2,000 and 200. Okay, and we just check they're in the right order as they should be in the answer, and that's fine. So then what we have to do is we have to find a common factor that they'll all divide into. There's one quick way of doing this, which is we look at the number of zeros at the end of them all, and they all have 2 zeros. So that means we can divide by 100 in all of them, and we'll very quickly get to this answer by just removing the 2 zeros at the end. Okay, so the answer would be 25, 20, and 2. Okay, so that's a little bit of a maths question, but hopefully that isn't too challenging for you. Explain why a muscle cell contains a large number of mitochondria. So muscles, we know, are involved in muscle contraction. So muscle contraction. Now, muscle contraction requires ATP, and ATP is our energy currency in the body. It's produced by our mitochondria. So there's a high energy demand. Okay, we get a high energy demand. So that would be enough to get you 1 mark for this question. It's worth 2 marks, you'll notice. And the second mark is for saying what I mentioned a moment ago, which is that the mitochondria is the site of ATP production. The mark scheme says you can get away with saying aerobic respiration. Okay, so something along these lines would be what you're needing for the 2 marks. Why does it have a large number of mitochondria? Because there's a high energy demand for that type of cell, for the muscle cell, because of muscle contraction, which we know requires ATP. And then we link it to a respiration topic where we know that mitochondria are the site of the second part of respiration, that aerobic part, and where we require oxygen. Okay, so that's your 2 marks. So let's have a look at question 2 here then. So the concentration of some ions within the cell sap of seaweed, the surrounding seawater, and a unicellular organism were measured. The results are shown in the bar chart. Okay, so we get three ions there, sodium, potassium chloride. The striped bar there is the cell sap of the seaweed. The central white bar is the seawater, and the right-hand shaded bar is the unicellular organism. Okay, fine. So part 1, says name the ion that moves from the seaweed into the seawater by diffusion and give a reason for your choice. Straight away, we're thinking diffusion, we're moving from a higher concentration to a lower concentration. So let's establish what direction that is based on the question. So it moves from the seaweed into the seawater. So the seaweed is the diagonally striped bar and the seawater is the white bar. So can you see any of the three ions where the diagonally striped bar is higher than the white bar? And immediately you can see that's going to be potassium as the ion. Okay, and what's the reason? Well, the reason is that there is a higher concentration of potassium in the seaweed and a lower concentration in the seawater. Okay, something along these lines. As long as you've given where the higher concentration or the lower concentration is, which indicates the direction of movement of the molecules, then you're going to get the mark for that one there. Next question, the unicellular organism must always have a sodium concentration of below 0.25 milligrams per litre. Name the process that moves excess sodium ions out of the unicellular organism. Okay, so let's look at sodium and the unicellular organism. Now, we can see from this shaded bar, it's at 0.15. Okay, so relatively low. The seawater around it has much more sodium in it. Okay, now by diffusion, sodium will be moving into this unicellular organism from higher to lower. So that's into the organism. So if it wants to move sodium ions out of itself, it will need to use the opposite method of transport, which is active transport. That's where we go from a lower concentration to a higher concentration. And lastly, the cell sap of the seaweed was tested to measure the concentration of each ion. Name the structure in seaweed cells that contains cell sap. So the cell sap is contained in the structure called the vacuole, and that's a fairly straightforward knowledge-based question for you to answer. Okay, question 3. The enzyme invertase is commonly used to make soft-centered chocolates. Sounds nice. Invertase speeds up the following reaction. Sucrose is catalyzed by invertase to produce glucose plus fructose. Part A, an investigation into the effect of pH on invertase at different temperatures was carried out, and the results are shown below. You can see them in the graph. Okay, so we've to identify the temperature at which invertase was the most active. Okay, so we've got two graphs here. We've got one showing 45 degrees, where the enzyme activity is significantly higher than at 55 degrees. So the temperature where it was most active is where there's the most enzyme activity, so therefore that's going to be 45 degrees. We've then to state the optimum pH of invertase, and again, if we look at both graphs, we can see the peak in enzyme activity is at pH 5 for both. So therefore we can happily say pH 5 is our answer there. 3A, part 3. Predict what would happen to the enzyme activity if the investigation was repeated at 75 degrees. Now, what it's asking you here is not what will happen to the enzyme, and a lot of students would quickly write down here the enzyme would be denatured, which is true, but the question is asking you to predict what would happen to the enzyme activity. So if the enzyme is denatured, the enzyme activity is going to either decrease or simply stop altogether. Okay, so there'll be less activity or no activity, less or no activity. Okay, so an answer along those lines would get you the mark for that one. The key thing is try and use the word activity when you're describing enzymes as often as possible. And when you're asked to predict what would happen, it's usually an increase or a decrease, depending on whether it's a temperature increase from a lower to a slightly higher temperature, or usually it's a decrease when you get in very high temperatures like 75 degrees. Part 4. Describe how this investigation could be changed to find a more exact optimum temperature of invertase. So this investigation was done at both 45 and 55 degrees. Now, the result of this was that 45 degrees obviously produces a much greater enzyme activity than 55 degrees. But what we don't know for sure is that if we had a temperature in between 45 and 55, would that in fact have been the optimum temperature? So what we'd want to do here is we would want to repeat, but this time with more temperatures, both above and below the optimum, which was 45, wasn't it? Okay, so we'd repeat with more temperatures both above and below the 45 degree. Okay, part B, a different enzyme speeds up the following reaction. Starch is broken down into maltose. Explain why invertase would not speed up this reaction. So the key here is to do with specificity of the enzyme to the substrate. We know that the active site has a particular shape on the enzyme, and that shape is specific to only one substrate. So in this case, invertase is specific, if we scroll all the way back up here, to sucrose. Okay, that's its substrate. So therefore, why would it not speed up this reaction? Because it is not specific to starch. Or you could say something like, it is only specific to sucrose. So like the inverted version of the answer there. Okay. And just if you wanted to improve that answer, you might say something like, rather than saying it is not specific, you would say the active site is not specific to starch. That would be a better answer. And similarly, where I've written it here again, you could say the active site is only specific to sucrose. So something along those lines would get you the mark for that particular question. On to question 4. It says, interferon is a type of protein made naturally by cells of the immune system. It can be manufactured by genetic engineering and is used to treat viral infections. So interferon was one of the first human proteins to be manufactured by transferring genes between organisms. And that diagram there should be familiar to most of you from your topic on genetic engineering, and hopefully you recognise all the steps there. But the first question is a definition question. It's asking you what is a gene, and from your notes you should know that a gene is a section of DNA that codes for a protein. Okay, section of DNA that codes for a protein. Okay, that's a simple memorization. You just have to learn the definition for the gene. Part 2, describe one of the uses of enzymes in the process of genetic engineering. So enzymes can be used to either extract or to, you could say, cut out the gene, or you could say it's used to insert the gene, okay? Or it's sometimes used to cut open the bacterial plasmid. Okay, and you don't need to know the technical ins and outs of that, but basically by using the same enzyme, you're more likely to be able to stick the gene into the plasmid. It will have complementary bases there, but you don't need to know all that at National 5. Okay, so that's one of the uses of enzymes, anything from that list there. B, part 1. Before it's manufactured by genetic engineering, interferon was extracted from white blood cells. 25,000 litres of blood were needed to produce 400 grams of interferon. Calculate how many litres of blood were required to produce 1 gram of interferon. Okay, so what we're going to do here is we know how many litres of blood are needed for 400 grams of interferon, but we want to know how many would be needed for 1 gram of interferon. So we know the total grams of interferon for both here. So we simply say that 45,000 litres of blood is 400 times more than what we need for 1 gram of interferon. Okay, 400 grams of interferon needs 45,000. 1 gram, which is 400 times smaller, is going to need 400 times less than 45,000 litres of blood. So we do a calculation, we do 45,000 divided by 400, and that should give us 112, 400, should give us 112.5, okay? And then we pop our answer in there, 112.5, okay? So 400 grams of interferon for the first one. If we want to know 1 gram, that's 400 times less. So we then divide the 45,000 by 400. Okay, B, part 2. During its manufacture by genetic engineering, 85 milligrams of interferon can be produced per litre of bacterial culture. Calculate the mass in grams of interferon produced in 45,000 litres. Okay, so if in 1 litre, because it says per litre at the top there, we produce 85 milligrams, we have to calculate the mass in grams of interferon produced in 45,000. So the first thing we do is we say, right, we can produce 85 in 1 litre, and it's asking us for 45,000 litres. We times the 85 by 45,000. And I'm going to have to use my calculator for that because my mental maths is not good enough to quickly do that. Okay, and that will give us quite a big number, 3.825 million. Okay, so a big number, but it also tells us that we want the answer in grams and that 1,000 milligrams is 1 gram here. So what we have to do is then we have to divide this number by 1,000. So when we divide that by 1,000, again, I'm just going to quickly do my calculator here. That would give us 3,825, okay? And we go 3,825 grams as our final answer, okay? So a couple of little stages in that calculation, but that's how we would work out something like that. Fortunately, they gave us the conversion there, so we knew we had to divide it by 1,000. Name another type of protein produced by the immune system in response to pathogens. Now, we know that our proteins are share, structural, hormonal, antibody, receptor, and enzymes. The type of proteins produced by the immune system, we should hopefully know from that topic, is the antibodies. Okay, that's again just a bit of knowledge there. And if you don't know your share for your proteins, then learn those as well. Not just what the acronym stands for, but also what the function of each of those proteins is. Okay, so question 5 is one of those questions where if you have done the hard work of memorizing your notes and your content and you feel confident going into the exam, this is the kind of question that should excite you, because you're basically given an open book to write down everything that you've learned about that particular topic. So this one says, describe the breakdown of glucose in animal cells when oxygen is present. So the hint you're looking for there is basically oxygen is present, animal cells. So we're thinking aerobic respiration. Okay, so you're going to write down everything you know about aerobic respiration. So the first thing is that glucose is converted into pyruvate. Okay, 2 molecules of pyruvate. So that would get you 1 mark. Then the pyruvate is broken down into carbon dioxide, and you can use the chemical symbol here, that's fine, that's acceptable, and water. Okay. Finally, the third mark for this first part is that we have energy being released. Okay. So that energy is released in the form of ATP. Okay. Energy released as ATP. Okay. And you could also add in here a large number. Okay, we don't need to give a precise number here. We just have to say a large number of ATP. If you wanted to say that in stage 1, as an alternative to this, you could say in stage 1, 2 ATP are produced, and you could actually say the value that's often quoted, which is 36 ATP being produced here as well, for a total of 38. But a large number is sufficient for your answer here. The other things that you could say are respiration begins in the cytoplasm, but for aerobic respiration, it's completed in the mitochondria. Okay, so it begins in the cytoplasm and is completed in the mitochondria. Okay, and I should say, and aerobic respiration, because although it does mention oxygen being present in the question, we should just make it clear to the marker that we understand that it's aerobic respiration. Okay, so that would also get you a mark. And the last thing to state is respiration is, and I get my classes to say this all the time, is an enzyme controlled series of reactions. Okay, so any mention of enzymes controlling this would also get you the mark there. Okay, so there's 5 points that you could put there. Any 4 of those 5 points would have got you the full marks in this question. Okay, so that's a nice one. I think I always quite like those questions because you can just show off your knowledge a little bit in them. Okay, number 6, we've got the diagram represents part of the human nervous system. We've got parts S and part T. Okay, the central nervous system, CNS, consists of the brain and one other structure, name the other structure. So common mistake here is for people to write down it's the brain and the spine. You have to remember it's the spinal cord. Okay, the spine obviously also involves like the vertebrae and the bone and so on, but the spinal cord we're looking for the CNS. The structures S and T in the diagram are parts of the brain used to name S and T. So S is the cerebrum. Okay, that's the part that controls all of our kind of conscious, voluntary things like our memory, our personality, our intelligence, etc. To give the function of structure T, we have to identify what structure T is first of all. So it's the medulla. And once we've identified it's the medulla, we then have to give the function of the medulla. The usual functions are it controls heart rate or breathing rate as well. Okay, you could also say something along the lines of it controls any involuntary or unconscious process within the body. That's not necessarily true, but it's true of some of those processes. And as I say, you get the mark anyway for saying controls heart rate and breathing rate, which is pretty much typically what I would teach my classes to write there. Part C, state the role of the receptors in the nervous system. So our receptors detect the stimulus. Okay, so the stimulus is like the sensory input. So for example, a stimulus can be a light or a sound or a movement or a change in temperature or something similar. Okay, any one of those would count as a stimulus, but the role of receptors, the job of the receptor is to detect the stimulus. Okay, on to question 7. The diagram represents some of the events that occur during reproduction and early growth in humans. We have to name the site of sperm production in the body for A. So sperm production in the body is the testes. Okay, that's a fairly straightforward knowledge-based answer. We tend to do okay on these reproduction questions as well, generally in class. So hopefully that's the same for all of you guys. Describe how the chromosome complement of a sperm cell differs from cell A. So cell A, we need to identify that first of all. It's the diploid zygote. And cell A, the sperm cell is a gamete, which means it's haploid. So we can either say sperm is haploid and the zygote is diploid. You could just say cell A instead of zygote there as well, okay? Or you could explain the definition for both of them. So you could say the sperm has one set of chromosomes, whereas the zygote, you can see that's a much longer answer, so I'd probably use the other one if I was you. But yeah, the zygote has 2 sets of chromosomes. You could be more specific than that if you happen to know like the chromosome number, for example. So sperm has 23 chromosomes. Humans have 46 in total for a diploid cell. So the sperm is 23 and the zygote is 46. You could also acceptable to say that the sperm cell has half the number of chromosomes than the zygote, and also acceptable to say the zygote has double the number of chromosomes as a sperm cell. So something along those lines would get you the mark. But I think that probably that first answer is the one I would always teach and recommend that people go with. But you've got a few options there. You have to name process B. Now, process B is going from the zygote to a little clump or ball of cells. Those cells are known as the embryo at that point. So the cell has to divide to become those 4 cells that we see in process B. And the name of the process which involves cell division that we've learned about in the course is mitosis. So you will also get the mark there if you say cell division, but mitosis is the name given to that process. Okay, next one, we've got early embryos contain stem cells. What feature of stem cells makes them important in the early embryo development? Okay, easy one here. If you just simply say they are unspecialized, okay, that's what I think most students would put here. But you could also talk about them having the potential to become lots of different types of cells. You could also talk about that they can self-renew, they can make more copies of themselves, and that they're involved in growth and repair. Actually, all of these would be acceptable from the SQA for this answer. But as I say, the first one that springs to mind there, and the one that I think I teach with the most emphasis, is they are unspecialized cells. And that does come up reasonably often, so it's well worth remembering that stem cells are unspecialized. Okay, we'll move on to question 8. Question 8 says the ability of a person to roll their tongue rolling again, we get this all the time in exam papers, is a dominant characteristic represented by the symbol capital R. So part 1, a male with the genotype capital R small r and a female with the genotype small r had a child who was unable to roll their tongue. State the genotype of this child. So it says tongue rolling. If we read the core of the question at the top there, tongue rolling is a dominant characteristic, okay, and it's represented by a capital R. So if a child is unable to roll their tongue, therefore they can't do it, they must have 2 lowercase r's, because if they have even 1 capital R, they will have the ability to roll the tongue because it's a dominant characteristic and you only require 1 allele for the dominant characteristic. Part 2, tick 1 box to identify whether the child's genotype is homozygous or heterozygous and give a reason why. So small r is homozygous, and the reason why is because both alleles are the same. Okay, that's the definition for our word homozygous. Both the alleles are the same. If the alleles were different, say a capital R and small r, we would say heterozygous, but in this case they're the same, so it's homozygous. Okay, the male and female are having another child. Predict the percentage chance that this child will be able to roll their tongue. Okay, so we've got a male who is capital R and small r, and we've got a female who is small r. So if we look at all the different combinations here, now you'd probably want to draw out a Punnett square here, ideally, but I'm going to be typing this, so I'm just going to write out what the combinations would be. So the father, the male, has a capital R. That means there will be 2 offspring with a capital R, okay? The mum only has small r's, so we'll get 1 there, and we'll get 1 there. We will also get the small r from dad for 2 more of the offspring there. And because, again, the mum can only give small r, we'll have that as our 4 offspring. So our 4 offspring, we've got 2 that can roll their tongue, 2 that have got capital R's, and we've got 2 that cannot roll their tongues because they've only got lowercase r's. So the percentage chance of this is 2 out of 4. So that's a percentage of 50%. Okay, and the last question here, state which type of variation is shown by tongue rolling ability. With tongue rolling, you can either roll your tongue or you can't roll your tongue. Okay, so there are 2 options there, which means that has to be discrete variation. Remember, distinct groups, distinct categories. You either roll your tongue or you don't roll your tongue. If it was continuous, we would see a wide range of values from a minimum to a maximum. But for discrete, we're looking for distinct groups, i.e. the ability to roll your tongue or not. Okay, on to question 9. So an investigation was carried out to find out which leaf surface loses the most water through transpiration. 3 plants were set up as shown. There was petroleum jelly used to prevent water loss. So plant 1 had no petroleum jelly in the leaves at all. Plant 2 had petroleum jelly only in the upper surface, and plant 3 had petroleum jelly only in the lower surface. The plants were weighed, then left for 24 hours at 20 degrees before then being reweighed. Question 1, knowledge question. Name the openings found on leaves that the water evaporates through. It's one of everyone's favourite words in biology, stomata, okay? Or this stoma as well would be acceptable there. Which plant was used as a control in this investigation? So remember, a control is where we have the absence of the thing we're investigating. So because we're investigating which leaf surface loses most water, we would have to have a control where neither leaf surface was covered in petroleum jelly, which means that has to be plant number 1. Suggests why it was necessary to cover the plant pots with a polythene bag. This has come up in a past paper before. It is to prevent the evaporation of water via the soil. Okay, via the soil. Okay, we put that polythene bag in there, and it means that if water does evaporate from the soil, it will be caught by the polythene bag and then drawn back in and being able to be used by the plant then. We only want water that is lost via the leaves, not water that is lost via the soil. So that's why it was necessary to cover that. D, we've got the results for the investigation shown in the table. So, we can see plant 1, which had no petroleum jelly, lost the most at 40% decrease in the mass. Plant 2, which had the upper leaves covered in petroleum jelly, lost 36% of its mass, and 3, which had the lower leaves covered in petroleum jelly, lost 4% of its mass. Now it asks us, why were the results calculated as a percentage change? Now again, this has come up once or twice before in past papers, and the answer is pretty much always the same. When you're asked to calculate a percentage change rather than the actual change, the reason is always the same, which is that the starting values are different. In this case, it was mass. Okay, so if the starting values are different, you have to use percentage change as a means to calculate the difference between them. The next one, with reference to the aim, give a conclusion for this investigation. So when a question says in bold, with reference to the aim, you need to go and look at the aim. The aim of this one is to find out which leaf surface loses the most water through transpiration. So therefore, our conclusion has to refer to that. So if we have a look at the results, when the upper leaf surface was covered in petroleum jelly, we still lost 36% of mass. But when the lower leaves were covered in petroleum jelly and blocked, we only lost 4% of mass. So that clearly tells us that the lower leaf surface is where most water is lost, okay, through transpiration. Because they mentioned that in the aim, didn't they, through transpiration. So just for completion, we'll write that. But actually in the marking schemes, it says in brackets through transpiration, so you don't have to necessarily say that. All right, so most of the water is lost through the lower leaf surface. That is our conclusion, because our aim asked us which of the surfaces would lose the most water. On to question 10 then. The results of a blood count for 2 patients are shown. So we've got the white and red blood cells, we've got the hemoglobin, we've got the platelets, and we've got the normal ranges. Then we've got what patient R and patient S both have in terms of those blood components. So which patient has all 4 of their components within the normal range? So if we look at the white blood cell, that's 4 to 11 for the normal range, and both patient R and patient S are within the normal range, so that doesn't help us. The red blood cell is 4.5 to 5.5, and we can see patient R is within that range at 4.97. However, patient S is not within that range at 4.01. So we can state now that it's going to be patient R, but we'll just double check the other ones. Hemoglobin is 130 to 170, and we can see R is within that, and S is not. And platelets is 150 to 450, and we can see again patient R is within that. Patient S also is there, but because they weren't within the normal range for two of the components, then we know it has to be R there for that. Next question, 4 weeks later, both patients had their blood retested. The hemoglobin content of the blood of patient S had increased by 20%. Calculate the number of units of hemoglobin in this second count. Okay, so S and hemoglobin, so we've got 115 if we read that off the table, right? Now we want to find a 20% increase. Quick way of doing that is to work out, okay, well, a 20% increase is the equivalent of 1.2, okay? Because one timesing it by 1 gives you 100%, but timesing it by 1.2 will give you an additional 20% on top of that. 115, I'll do this in my calculator, 115 times by 1.2 times by 1.2 gives us 138. Okay, and that is our answer there, 138. Now, another way to work that out, if you're not as confident about just adding a percentage as a decimal, is to say, well, if we want to increase it by 20%, then what we need to do is work out what 1% is, and then add 20 of those on top of it. So 115 divided by 100 would give us 115 divided by 100 would give us 1.15, and then we need to times that by 20. Times that by 20 would give us 23, and then we need to get the original, which was 115, and add that 23 on top of that, and that would give us 138. So there's a couple of ways of working it out depending on which way you're more mathematically comfortable with. Okay, part B, other than containing hemoglobin, describe one way in which a red blood cell is specialised to carry out its function and explain the advantage of this specialisation. Okay, so there's a couple of things that we could say here. We could say that it has no nucleus. The advantage of no nucleus is it gives us more space to carry more oxygen. So that's one option for you. You could talk about the biconcave shape, and the function of the biconcave shape is similarly to increase the surface area, so larger surface area. And you'll notice this answer coming up lots in biology, the old large surface area. And what does that mean? Well, that means that we can carry more oxygen or transport more oxygen or absorb more oxygen. Okay, so more oxygen is carried to wherever it needs to go. Okay, so something along those lines would have got you the mark for that one there. And see, I like these questions. These are ones that we've seen before a few times in past papers, and we certainly practice this quite a bit in class. So again, it's a good one for students when these come up. Two types of blood vessels are arteries and veins. Compare the structure of these two vessels by these two types of blood vessel. Now, the easiest thing to do here is, because it's a comparison question, is to say arteries have this, whereas veins have this. Arteries have this, or veins have this, but arteries do not, or something similar like that. So we would say something like arteries have thick muscular walls, right? Whereas the veins have much thinner walls and less muscular. Okay, so there you go. That would get you 1 mark. You could say that the veins have valves, whereas the arteries do not. And notice how I'm always saying whereas, and I'm making it a comparison statement. You could say that the arteries have a narrow central channel, whereas the veins have a wider central channel. Okay. So something along those lines would be good enough to get you the mark there. And if you get all three, you get all 3 marks. Okay, folks, so on to question 11. This is our scientific literacy question, where you get an excerpt from a scientific research report, and they give you a little taste of that, and you have to do something with the information they give you. So let's read the info first. So in a study, scientists interviewed 350 British athletes competing in different sports, including cycling, swimming, rowing, athletics, football, hockey, and rugby. Approximately half of these athletes have tooth decay compared to a third of adults the same age. The study showed that these athletes put more effort into looking after their teeth, with 95% of them brushing their teeth twice a day compared to 75% of the general public. Only 21% of the general public flossed their teeth regularly compared to 44% of the athletes. The study also found that smoking rates in the athletes were found to be lower and overall diets were better compared to the general public. However, 88% of elite athletes used sports drinks, 56% used energy bars, and 72% used energy gels frequently during training and competitions, said one of the researchers, adding that the sugar in these products increases the risk of tooth decay and their acidity increases the risk of tooth erosion. The scientists were surprised to find that elite athletes have more tooth decay than the general public. Use information from the passage to explain why this was unexpected. So if we look at the passage, why is it unexpected that the athletes have more tooth decay? Well, for example, they are far more likely to brush their teeth. Okay, 95% of them are brushing their teeth twice a day compared to 75% of the general public. Only 21% of the general public floss their teeth compared to 44% of athletes. So something along these lines would be an acceptable answer here. Something like athletes brush their teeth. They brush their teeth twice a day. I should say athletes are more likely, because many of the general public do, but it's just there's a higher percentage of athletes doing it. You could say that they are more likely to floss more regularly, or something similar to that. The SQA also accept something like the athletes smoke less, which is in the third paragraph there, or that they have better diet, again in the third paragraph there. Any one of those answers would be acceptable for that one. There's quite a few to choose from in the passage. Calculate how many athletes flossed regularly. So for this one, we have to find out how many floss regularly, and it was 44%. And the total number of athletes was 350. So our calculation is going to be 350 times by, if we change 44% into a decimal, it's 0.44. So 350 times 0.44 is 154. The other way to do it, if you don't like changing your percentages into decimals, is to do 350. And then we find 1%. So we divide by 100 to do that. And that gives us 3.5. And then we times that by 44 to get 44%, and 3.5 times 44 by 4. Okay, so either way is acceptable to work out the percentage there. Part C suggests why this study may not be valid. So an emphasis here, you can see in bold, this study may not be valid. Why is this particular study not valid? Well, when it comes to tooth decay, there are so many variables involved, okay? There are too many variables involved. So many of them are not taken into account okay? You could say something like it was impossible for the researchers to control all the variables, or it was very difficult for them to control all the variables, or tooth decay could be caused by other things than the variables that were accounted for in this study. Okay, so something along those lines. But remember, whenever your question talks about validity, it's VAVA, so valid variables. So your answer should include variables, okay? Suggests why this study may not be valid. It's because there's too many variables that can cause tooth decay. Okay, 11D. The table shows the nutritional content of a sports energy bar, Energy Blast. ATP. You'd be lucky to find a sports bar for 80p in a shop these days, but never mind. We've got the content as well there. We've got the fat, sugar, protein, and other content per 100 grams of the bar. So you have to use the information in the table to complete a pie chart. Now, often in the exam, you have to do a bar chart or a line graph, but it's unusual for you to do a pie chart. However, it is a skill that I do get my students to practice, and that you should practice for your exam just in case you get asked for it. So the first thing you need to do is you need to work out that the pie chart has got 20 little segments, and because there are 20 segments on the pie chart and the total mass is 100 grams, that means 100 divided by 20, that each section is worth 5 grams. So for example, the other section, which is 10, would be 2 sections or 2 parts of the pie, because each part's worth 5. 2 times 5 is 10. Protein, we divide 35 by 5, that gives us 7, so there should be 7 segments for protein. Sugar is the same, it's 35, so that again, that'll be 7 segments of the pie chart. And fat at 20 divided by 5 gives you 4 segments for the fat. Fat should have 4 segments, sugar 7, protein 7, and other 2 segments. Make sure that you have labelled each of the sections of the pie as well with the labels that are in the table. So fat, sugar, protein, and other should be labelled within the pie chart. And make sure you use, obviously, a ruler to draw straight lines and make them as accurate as you can to ensure you get the mark for this one. Okay, we're doing well. We're on to question 12 now. The diagram shows part of a food web in a pine forest. So the part A, complete the table by giving one example of each type of organism from the food web. Okay, so producers, we will find them at the bottom of the food web. They are plants or photosynthetic organisms that are at the bottom of the food web. So our examples here are either the pine tree or we could say the moss. Okay, that's where you find those. The predators. Now, a predator is an organism that hunts and eats another organism. Okay, so from this particular food web, we've got the fox, we've got the owl, the wood mouse as well, which is the predator of the earthworm, and also the hedgehog. Okay, not a renowned predator, I don't think. Is the first one that would spring to your mind if someone asked you for a predator. But yeah, there you go. According to our food web, the fox, the owl, the wood mouse, or the hedgehog would be acceptable for that one. You've to use the word increase, decrease, or stays the same to suggest what might happen to the squirrel population if all the foxes were removed. Okay, that's a common question that comes up quite a bit. So if we remove the foxes from this particular, what is going to happen to the squirrel population? Now, the squirrels are predated upon by the fox. So without that predator there eating them, the squirrel population would increase. Okay, so we've got an increase. Now, that's the easy part. The difficult part is the explanation for that. Why does it increase? Well, you've got to say something along the lines of no squirrels will be eaten by the foxes, or there are no foxes to eat the squirrels would be acceptable as well. You can say fewer squirrels are eaten by predators, or there are fewer predators to eat the squirrels, or something similar. Now, this is one of the rare times where, so that I think that's the most straightforward answer, and that's the one that I would suggest. Not very good with my spelling of squirrels there, am I? That's the one I would suggest that you go for. However, there are other acceptable answers here as well. If you said that the squirrel population decreased and your reason for it was that there's less competition or more food for the owls, so the owls eat more squirrels, then that would be acceptable. Similarly, if you said the squirrel population stays the same, and the reason that you said that was because of a combination of both of the actions, so i.e. it would increase because there's fewer foxes, but it would decrease because there is less competition for the food, so therefore the owls would eat more squirrels, then that would be acceptable as well. Okay, part C, state the term used to describe the role of an organism within its community. One of our favourite words in biology, sounds great, is the word niche. So the niche is the role of an organism within its community. Part D, the table shows some features of fungi commonly found in Scotland's pine forests. So we've got our 5 different types of fungus, and we've got 3 attributes: the maximum cap size, the cap colour, and the stem colour. So we're going to go through this paired statement key, and we are going to fill in our answers based on the information we're given. So paired statement 1, stem colour is not dark brown, so we're going to go to 2. Okay, and if it is dark brown, we're going to go to 3. So let's look at the ones that are not dark brown first of all, okay, for the stem colour. So that's going to be the sycamore, which is white, and the pinewood gingertail, which is brown. Now, when we go to part 2, the sycamore's already in there, which means this must be the pinewood, great names, right, gingertail, okay. For the statement go to 3, they were the ones where the stem colour was dark brown. That was our other 3 here. And statement 3, paired statement is cap colour is dark brown. So who's dark brown? We've got the ear pick fungus and the Pelargonium bonnet, and we've got the velvet rollworm, which is the one that's mid brown. So because we've got 2 that have a cap colour of dark brown, that means we have to go to 4. Because we've got 2 options there, so therefore we have to go to another paired statement. And this time the cap size is 1 centimetre, that's going to be the Pelargonium, and the one which has a maximum cap size of 2, which is still left because obviously the pinewood gingertail is also 2, but the only one that's left and has followed this sequence is the ear pick fungus. Again, a cracking name, isn't it? Ear pick fungus. So there you go. That's how we work through our paired statement key for that question. Okay, next question is a photosynthesis question. Almost certainly guaranteed to get one of these in every exam. An experiment into photosynthesis was carried out. After the lamp was switched on, the time taken for the leaf disc to float to the surface was recorded. The experiment was repeated 5 times and the results are shown in the table. Fantastic. Okay, we got 5 times there for the disc to float to the surface. Calculate the average time taken for the leaf disc to float to the surface. So what we're going to do here is we are simply going to add all of our values together. So that's 18.4 plus 17.8 plus 11.7 plus 13.8. And if we add all them into our calculator, we'll get a value, and then we divide that value by 5, and our calculator should show 15.66, or if we want to round it up and be neat, we could say 15.7. That would be acceptable as well. Okay, 15.7. So we add up all 5 numbers and then we divide by 5 to get our answer there, which is 15.7. A gas produced during the first stage of photosynthesis diffuses out of the cells, causing the leaf discs to float to the surface. What is the name of that gas? Straightforward knowledge question. Light reaction stage. The gas that's a waste gas that's diffused out of the leaf is oxygen, so we pop that in there. Okay, during photosynthesis, the light energy from the lamp is converted into chemical energy. Name the molecule generated by this chemical energy that is then used in the second stage of photosynthesis. So again, this is a knowledge-based question. You should really have learned this, and it's a molecule that's quite common across the course. We talk about a lot, which is ATP. Okay, the second stage of photosynthesis is affected by temperature. Name this stage and give a reason. So the second stage of photosynthesis, again, it's a knowledge-based question, is carbon fixation. Now, this is a bit of a trickier question. What is the reason? Why is it affected by temperature? Well, we know, like respiration, that photosynthesis is an enzyme-controlled reaction, or a series of enzyme-controlled reactions. And because of that, because it's controlled by enzymes, it's going to be affected by temperature. So enzymes are affected by temperature. Just to complete our answer and make sure of getting that mark there. Question 14. In an investigation into the distribution of woodlice in a woodland, students collected and counted them every day for 5 days using the sampling technique below. Hopefully you all get a chance to do this at some point in the course, and you go out and you set one of these and you found some invertebrates, but this is a pitfall trap. Okay, identify 2 variables that should be kept constant when setting up this sampling technique. So there's a number of different things that you can choose from here. The size of the trap is probably an important one. The width of the trap as well. The shape even, that should be kept consistent. The depth of the trap, where it's level with the surface. The number of leaves that you put on top as well, that has to stay consistent. Basically, anything which could affect the number of invertebrates that would go into the trap has to be kept constant. Okay, so there's quite a lot of different things that we could have with there. Okay, the SQA accepted all of those options. Part B, state one way in which the results could be made more reliable. Okay, so we've set a trap, we've done it over the course of 5 days, wasn't it? Okay, how could we make the results more reliable? We could set more traps. Okay, that's simple repetition, isn't it? Okay, the SQA also allowed you to say repeat the experiment or investigation, which is a bit of a general term, isn't it? Experiment, investigation, rather than being more specific, which is saying to set more traps. But they accept both of them there, so you can use either of those 2 to get the mark for that one. Okay, question 15, we're doing well now. The diagram shows the transfer of energy through a food chain in an ecosystem. The numbers represent the energy in the different populations. So what we have to do is we have to name the type of diagram shown. Now, it mentions it's showing the transfer of energy and it's in the shape of a pyramid. So this is quite a straightforward one. It is a pyramid of energy. Part B, 5% of light reaching the plant plankton and is converted into new plant material. Calculate how much energy the plant plankton received. Okay, so the plant plankton, not so easy to say fast, has 42,000 units. Now we know that's 5% of what it actually received. It tells us that in the question. So we have to do a reverse percentage calculation here. So what we want to do first of all is find out what 1% is. If 5% is 42,000, what is 1%? So we do 42,000 divided by 5, okay? And I'm just going to quickly pop that into my calculator to make sure I get this right rather than doing it mentally. So 42,000 divided by 5 is 8,400. Okay, so 1% of energy is 8,400. If we want to find out the total energy, i.e. wouldn't, we then have to times that 8,400 by 100. Okay, we times that by 100 and it gives us 840,000. Okay, 840,000. 84, okay, folks, that's how you work that one out. State one way in which energy can be lost from a food chain. Well, you've got a choice here. You could say heat, you could say movement, and the one we don't talk about as much, undigested materials. Okay, so any one of those three. You don't have to put all three to get the mark for that one. Question 16. A wren is a small bird found in most parts of Britain. Wrens found in the St Kilda Islands off the west coast of Scotland differ from those found in the Scottish mainland in several characteristics, including colour, size, and wing length. It's thought that the St Kilda wren is evolving into a new species. Suggests how scientists could prove that the St Kilda's wren is not yet a different species from the Scottish mainland wren. So when you see a question that starts off suggests, you know it's going to be a slightly tougher question, sort of the A-level type questions from the exam paper. However, you should have been taught what the definition of a species is, which is that it has the ability to interbreed and produce fertile offspring. So what could they do? Well, they could interbreed the ones from St Kilda and the Scottish mainland and see which ones produce fertile offspring. If they produce fertile offspring, they are still the same species. If they can't produce fertile offspring, they are no longer the same species. So that's how they could prove that it's not yet a different species. When they interbreed, they would produce fertile offspring. Okay, part B, we have to put these series of statements into the correct order. Okay, they're not in the correct order here. This is the events that are go along in the sequence of the evolution of a new species. Okay, so first up, part of the mainland population would become isolated. Okay, so an isolation barrier is the first step. The second step is the mutations would occur within each subpopulation, so that's going to be D. The next one would be that the natural selection due to different selection pressures is going to occur. Lastly, because A is already given for us, we know that E must come next, which is over time each subpopulation becomes genetically different. And then finally, the last step, a new species is formed. So we have to explain for C how natural selection results in the evolution of a species. Now, natural selection is also called survival of the fittest. But what does it mean to be the fittest? Well, it means you are the best adapted individual within your population. And if you're the best adapted, you are most likely to survive, to reproduce. And if you can survive and reproduce, then you're going to pass on your favourable alleles to the next generation, to your offspring. Okay. Or the SQ also allow you to say something along the lines of the number of favourable alleles will increase in subsequent generations or populations, or they'll increase in frequency within subsequent generations or offspring. Okay, so that is the end of section 2 of the 2025 past paper. I hope that's been helpful to do a little walkthrough of the paper and to go over the answers and to explain as much as I can with that. Okay, good luck with your studying and your exams and your past papers. Thanks, folks.